Here in part 2 of Unit 4, we're going to address the manner in which the body physiologically manages glucose within the carbohydrates that we consume. And I want to begin by discussing an index, a measure of the quality of the carbohydrates in the food we consume, and that's called the glycemic index. And the glycemic index is a number that was developed that characterizes how quickly carbohydrates, or rather glucose, increases in the cardiovascular system soon after consuming some carbohydrate. So it's a measure that doesn't have units. And once we consume carbohydrates, that the glucose levels in the blood will increase. But the question here is, how much will it increase and how fast will it increase? And the glycemic index is a number that's designed to give us a sense of that. There are two different standards. One slice of white bread or an oral glucose solution are both used as the 100% standard. And if we take a look at the bottom figure here we see a number of sample foods where we have a very high glycemic index. On the very high end, we have white bread. We see that a baked potato, russet potato, is also very high on the glycemic index. And these foods, being very high with a high glycemic index, suggest that if we consume that food, our blood glucose levels would rise very, very quickly. Note that on the low end, we have peanuts, soybeans, cashews, cherries, just some examples of low glycemic index foods. These are carbohydrate-containing foods, but if we consume them, the speed in which blood glucose increases will be much lower here than it would be on the very high end. So there are certain factors that affect the glycemic index of foods and basically how fast blood glucose would show up in the cardiovascular system soon after ingestion. And the first factor is the proportion of the carbohydrate content of the ingested food. Recall that there are three macronutrients: carbohydrates, lipids, and proteins. And within a particular type of food, the greater proportion of the carbohydrates among the three macronutrients, that will have the effect of increasing the glycemic index, and hence the appearance of glucose in the cardiovascular system would be faster. If we take a look at just some examples of different foods and their glycemic indices, we're going to compare it to a pure glucose ingestion. Ice cream has a glycemic index of 61, not on the highest level of the glycemic index, but still considered to be on the high end of a moderate glycemic index. A boiled potato is on the high end at 85, and instant rice, this is not brown rice, this is white rice, is also very high at 91. And in these two latter examples, almost all of a very high proportion of these foods are composed of carbohydrates as compared to lipids and proteins. And so being almost all carbohydrate, the starch component is very easily broken down and absorbed by the digestive system, and the appearance of glucose in the cardiovascular system would be very, very fast. A second factor that affects the glycemic index of foods is the type of carbohydrate. Recall that we have three types of dietary carbohydrates. We have sugars, starch, and fiber. Another classification is that the sugars would fit under the simple carbohydrate, while the starch would fit under the category of complex carbohydrate. And so consuming a simple carbohydrate or food that contains a high proportion of the sugars or simple carbohydrate would result in that food having a higher glycemic index. A complex carbohydrate takes longer for the digestive system to break it down, and therefore it would have a lower glycemic index. And one of the concerns with consuming a high glycemic index food is the role that it plays indirectly with fat storage. One of the effects of insulin is that insulin will directly promote the removal of carbohydrates from the blood and move into skeletal muscle and liver tissue, but indirectly insulin will also have a fat storage effect on adipocytes or fat cells. And so when we think about the high glycemic index of a food, it doesn't just affect the carbohydrates circulating in our body, it also tends to have an indirect fat promotion effect. Cooking will also, of one food, will also affect the glycemic index of that food, because cooking is the application of a form of energy in heat to that food, and that heat overcomes the strength of the chemical bonds holding the glucose molecules together. So heat will disrupt those chemical bonds, and that will lead to faster digestion once we've consumed that food. And if cooking makes it easier for our digestive system to break that carbohydrate down, we will be able to absorb the glucose molecules much quicker. And as an example here, if we take a look at the glycemic index of pasta that's boiled for 5 minutes, it's on a moderately low level at 34. But increasing the time to which we boil that pasta, doubling it to 10 minutes, we see it has the effect of increasing the glycemic index. So it doesn't move the glycemic index into a high level, but it's nevertheless, cooking still will break up the bonds holding the glucose molecules together in the starch of pasta and increase that glycemic index. A second measure that is often used to characterize the carbohydrates in food is something called the glycemic load. So where the glycemic index will have the effect or characterizes how fast blood glucose increases, the glycemic load also takes into consideration the glycemic index of the food, but it also takes into consideration how much carbohydrate exists in that food source. So as a numerical value, the amount of carbohydrates is multiplied by the glycemic index of that food, and it's divided by 100%. And we'll take a look at a couple of example values here. The glycemic load is an important measure, as it's been found to be a significant predictor of the development or the risk of developing type 2 diabetes mellitus and cardiovascular disease. And we will illustrate the effect of the glycemic index and the glycemic load with an example. And we'll compare a corn muffin with a bran muffin. So if we take a standard corn muffin, it has a glycemic index, a very high glycemic index of about 102, and this is compared to glucose, so a very high glycemic index. Corn, the starch in corn is all glucose, so it will have a high glycemic index. And per serving, 29 grams of carbs are delivered, yielding a glycemic load of 30. If we compare that to a bran muffin, we see that the bran muffin has a glycemic index that is about half of that of the corn muffin, because the bran muffin, the carbohydrates in the bran muffin are not all starch. A significant component of that carbohydrate is in the form of fiber, which we do not have the ability to digest. So the amount of glucose molecules absorbed will be lower. We have a lower amount of carbohydrates per serving in that bran muffin, owing to the fact that the carbohydrates are split between the starch and the fiber component. And we see that the glycemic load is about half of that of the corn muffin. So we have to consider the glycemic index, and there's less carbohydrates in terms of what is absorbable by the digestive system. that will lower the glycemic load. And if we're going to illustrate the effect of the glycemic index and the glycemic load simultaneously on blood glucose concentration. So along the y-axis, we're going to measure the level of blood glucose following the ingestion of two different food sources over time. And our first graph here illustrates the effect that if we consume a meal, and of course this meal has to contain carbohydrate in it, over time, as the digestive system breaks up that carbohydrate and starts to absorb the glucose molecules, we'll see blood glucose levels rise, they'll eventually peak, and they'll eventually come back down to pre-meal levels. Now, if we compare a second food, here we eat a different food, so the blue dotted line represents a different food, and we consume a serving of that food, and we see that blood glucose will increase, it will hit a peak, and it will come back down eventually to pre-meal levels. So the pattern here is going to be the same between these two food sources. However, the slope and the magnitude characterizes these two different foods. So if we imagine how steep these two curves are, the steepness of these two curves is reflected in the glycemic index. And recall, the glycemic index is that measure of how fast blood glucose increases in the blood soon after consuming a meal. But what's also very important here is how high blood glucose levels increase, and that's going to be reflected in the glycemic load. So the glycemic load as a calculated value, yes, it also considers the glycemic index, but this graph illustrates the fact that when we think about the glycemic load, it's not just how fast, but how much carbohydrate is delivered in that food source. So this is a complex illustration, but nevertheless very important to understand how our body physiologically deals with the glucose molecules after absorption. So here, what we have illustrated up at the top, this is a blood vessel that is transporting blood and eventually glucose molecules following a meal. The rest of this illustration is representative of a muscle fiber membrane. So here we see the membrane of a muscle fiber. Here we see the interior of a muscle cell, and we have the extracellular space. Embedded within the membrane of the muscle fiber are transport proteins that are specific to glucose. And that is what we call GLUT1. So GLUT1 stands for glucose transporter. It's a protein permanently embedded in the membrane of the muscle fiber, number one here, that will move glucose into the cytoplasm of a muscle cell. So if we start here at the very top, here we have blood flow moving from left to right, and we have a glucose molecule that is coursing through the cardiovascular system that will eventually move out of the cardiovascular system and will bind to asparagine 45. Asparagine 45 is an amino acid component of this protein that is the 45th amino acid from this end. So if you count up all the amino acids, number 45 is this particular one right here. Glucose will form a chemical bond with this amino acid, and this transporter will then move glucose into the cell of this muscle fiber, into the cytoplasm, where the cell will either store it as muscle glycogen or can use it as a source, as a fuel source for energy. Now, we've just consumed a meal, a carbohydrate meal, and we quickly see many glucose molecules showing up. But when we have many glucose molecules showing up, it exceeds the capacity of GLUT1 to manage glucose. But we also know that when we consume the carbohydrate meal, those glucose molecules will trigger the pancreas to release insulin. Insulin is a protein that functions as a hormone. So when the pancreas releases insulin, insulin will course through the cardiovascular system and will eventually bind to a very specific protein receptor in the muscle fiber, the skeletal muscle fiber membrane. That effect will activate enzymes on the interior of the muscle fiber, and it will cause another transport protein we call GLUT4. GLUT4 is the exact same protein structure as GLUT1, except that GLUT4 is stored inside the cytoplasm and only used when necessary. But when insulin is released, insulin binds to its receptor and it calls up additional transport proteins in the membrane where the glucose, all those extra glucose molecules can now be moved into the cytoplasm, bringing glucose levels in the blood down. So this is the normal mechanism that takes place when we have a very low amount of glucose, just slightly above resting levels present in the cardiovascular system. GLUT1 is going to be responsible for moving glucose into the cell, but following a meal, when we need much more help to manage all these extra glucose molecules, insulin will be triggered to be released by the pancreas, which will call up additional protein to move those glucose molecules into the cell. So in terms of the two very important pancreatic hormones in giving us the ability to physiologically handle our blood glucose levels, the first one here is insulin. Insulin is produced by what are called beta cells of the pancreas. And insulin, as illustrated on the previous slide, helps to take glucose into liver cells and skeletal muscle cells from the blood, thereby decreasing blood glucose levels. So once that glucose enters the muscle cell and the liver cell, it is converted into what we call glycogen. Nothing happens to the structure of the glucose molecule. It's just chemically bound to other glucose molecules. And the important point to take from this slide is that insulin has the overall effect of lowering blood glucose levels, and we do that following a meal. The structure of insulin here, it is a protein made up of 51 amino acids with two chains, A and B. We'll talk about protein structure in a subsequent unit, but this is certainly not to be memorized. But this gives us a pictorial illustration and idea of what this hormone looks like. The opposite acting hormone also released by the pancreas, except that this hormone is released by alpha cells, is glucagon. So glucagon is also released by the pancreas, but by different cells. It is also a protein that functions as a hormone, and it is made up of 29 amino acids. And glucagon is secreted when we are in a physiologically starved state. And so that would be realized by a very low level of glucose in the cardiovascular system. So if blood glucose levels drop below resting levels, the pancreas will detect that, trigger the alpha cells to release glucagon, and glucagon will find cells in the liver and trigger those cells to break up liver glycogen, break up the bonds between the glucose molecules, and release the glucose molecules into the cardiovascular cells. And into the cardiovascular system, making it available to cells in the body. Glucagon also has the effect of triggering a process called gluconeogenesis. we have the new formation, okay, so the new formation of glucose from non-carbohydrate sources, primarily amino acids. So we can form new glucose from amino acids, but we do so when the body senses that it's in a starved state. So combined with breaking up liver glycogen, combined with triggering cells to produce new glucose from muscle protein, i.e. amino acids, the effect here overall is to raise blood glucose levels. So this ends part 2 of Unit 4, and we'll continue in part 3 of Unit 4 by examining what happens when this normal physiological pathway involving insulin, primarily insulin, and carbohydrate ingestion is damaged by disease.